Good evening, everyone. On behalf of the Discovery Institute Center for Science and Culture, Colorado chapter, we'd like to welcome you all here to hear Dr. Behe tonight. We're very excited to have him. It's a little hard to get on his calendar, let's just say. So we're pretty excited to hear what he has to say about his new book. For those of you who aren't familiar with who we are, the Colorado chapter of the Center for Science and Culture brings speakers and has other events mostly so far in the Front Range of Colorado. So if you find this kind of event interesting and you want to know more, we have an information table in the back. You probably all saw it as you came in. We have a brochure, it's a fine brochure with the mountain goat on the front. This differentiates us from the Texas group, who has no mountain goats. So feel free to pick one of these up on your way out and read up on us and what we're about. We also have in the back a bunch of sample books. Now, I don't know about you, we don't have any for sale here because apparently the state of Colorado thinks we should collect sales tax and we don't have a license. So, I don't know about you, but I like to leaf through a book before I decide to buy it, kind of look at the table of contents and look at the chapters and decide whether it looks like a good read. And so we do have samples of all three of Dr. Behe's books back there. We also have books from all of our previous speakers, Marcos Eberlin, Dr. Doug Axe, and let's see who else, Dr. Mike Keyes. We also showed a film called Human Zoos, if you're interested in the cultural side of this debate. That's actually available, I think, on YouTube, so you can watch it for free now. So feel free to check that out. Anyway, without wasting any more time on introductory material, I'd like to invite Paul Ernst to come up and introduce Dr. Behe.
Good evening. In his Ode to Materialism, A Free Man's Worship, philosopher Bertrand Russell said the only sure foundation on which you build your life was a foundation of total despair. Christians have many reasons to know that isn't true, but according to a survey by the Discovery Institute, for about 50% of young adults, this depressing worldview dominates. Its principal support, even for the non-scientific, is Darwinism or evolutionary naturalism. Though dissent from Darwinism in its modern form began in the 1960s, it was Dr. Michael Behe's book, Darwin's Black Box, that exploded on the scene in 1996 and launched the intelligent design movement in its modern form. Intelligent design is the idea that some things in nature are better explained by intelligence than by the working of natural forces plus chance. It is not a religious idea, but a simple intuition that is backed up by the most recent science. In Mike's formulation, when you see a composition of matched parts performing a function, you are entitled to make a design inference unless it can be shown otherwise. And I might add, we are still waiting. If ID happens to have religious utility, that's the other guy's problem. Working strictly from observation and searches of the scientific literature, Dr. Behe concluded that there is no gradual, stepwise, that is Darwinian pathway to build the machines that operate the cell. These molecular machines, like his famous mousetrap illustration, are what is known as irreducibly complex. All the parts must be present with assembly instructions, or there is no function to select, and Darwinism is dead on arrival. As a new Christian and former science teacher, I read Darwin's Black Box with excitement as well as apprehension, as I questioned if this idea would survive the waves of criticism from the scientific establishment. I think it's safe to say that despite a few bumps in the road, it has stood the test. To introduce a new book, his new book, Darwin Devolves, I can't do any better than to read from the back flap. While Stephen Colbert has called Michael J. Behe the father of intelligent design, Behe's arguments have been called close to heretical by the New York Times Book Review, and Richard Dawkins has publicly taken him to task for his maverick views. Wherever he goes, Behe makes waves, but he has remained singularly focused on doing rigorous scientific analysis that points to controversial but incredible results that other scientists won't touch. This research has led Behe to challenge Darwin's theory of evolution in his seminal bestseller, Darwin's Black Box, arguing that science itself has proven that intelligent design is a better explanation for the origin of life. Now, 20 years later, Behe shows that new scientific discoveries made possible in the last few decades point to a stunning fact. Darwin's mechanism works by a process of devolution, not evolution. The mechanism works by breaking down genes, which means that evolution can make something look and act different, at least on the surface, but it doesn't have the ability to create anything at the genetic level. As I finished Darwin Devolves, I thought of it as an appropriate bookend to his work. Darwin's Black Box was about what natural process can't do. His second book, The Edge of Evolution, probed the limits of successive mutations. And Darwin Devolves tells us what evolution can do, and the answer is not much. Dr. Behe received his PhD in biochemistry from the University of Pennsylvania and did his postdoctoral work at the National Institute of Health. He is currently a professor of biochemistry at Lehigh University. So, on behalf of the Colorado chapter of the Center for Science and Culture, please extend a warm welcome to Dr. Michael J. Behe.
Thanks very much. Thanks very much for that very kind introduction. It's wonderful to be here with you. This is, I think, my fourth or fifth time in Colorado, and it's always just, very nice. The beautiful mountains. Not as tall as we have in Pennsylvania, but they're nice. They're nice. So I titled my talk, "Less is More: How Darwinian Evolution Helps Species Adapt by Breaking Genes." And it's based on very new research, past 10, 20 years of research in biology. And as we know from the history of science, science advances very quickly, oftentimes, when a new piece of technology becomes available. For example, when Galileo took a telescope and looked at the sky, our knowledge of the universe increased tremendously. When the microscope was invented in the early 1600s, a whole new world of life became apparent. Things that nobody had suspected even existed. Well, it turns out that we are living through a similar period. Because of the advance of new technology in biology, we are getting a view of life that nobody could have seen only a few decades ago. And that's the topic of my new book, Darwin Devolves. And in it, I'm trying to make 3 points. Something called the first rule of adaptive evolution, another point called the principle of comparative difficulty, and the family line. But because of time constraints, we'll just look at the first point, the first rule of adaptive evolution. So you'll still have to buy the book, okay? I got kids, okay? And the first rule of adaptive evolution began with the work of this man. This is Richard Lenski, who is a professor of microbiology at Michigan State University. Let me just see if it works up here. And about 30 or so years ago, he began a long-term experiment in which he grew bacteria in his laboratory. He took a flask of nutrient broth, actually took 12 flasks so that he'd have 12 replicates of what he was doing. And in a flask, he put a little bit of the bacteria E. coli, which is a common laboratory test organism and lives in the human gut. And there was enough food in the form of sugar, there was enough food in the flasks to allow the bacteria to grow overnight. And in that, it would go through 6 or 7 generations, then it'd run out of food and stop. And he would come in the next morning and take a small portion of that growth from the flask and put it in a fresh flask with new nutrient broth. And for a day, it would grow another 6 or 7 generations. And it'd come in the next morning, take a bit of that and put it in a fresh flask. And again, and again, and again. And he did this day in and day out through decades. He and a whole parade of students and associates going through his laboratory. So that now, the number of generations that line of bacteria has undergone is on the order of 70,000. 70,000 generations. And because bacteria are so small, you can grow a lot of them in a small flask. It turns out there have been trillions of bacterial cells that have been born and died in Richard Lenski's experiments. So he's got the large numbers of generations, large numbers of individuals to make pretty strong statistical conclusions on his work. And he did this, his rationale was just to see how they would evolve. What would they do? What would come along and what would become of them? And within a few weeks of his beginning that experiment, he saw that some helpful mutations had come along. That is, the bacteria had started to grow faster. And the ones that started to grow faster would reproduce more quickly, leave more offspring, and then crowd out the other bacteria, so they took over the whole flask. And then you'd see another mutation would come along and do something similar. Maybe another one would come along. So he was very excited. This was exactly what you expect of a Darwinian process. So he could study this in his laboratory. But the thing is that mutations are changes at the molecular level of life. They're changes in DNA and then the proteins that they code for. So in order to tell what exactly is going on, why are these bacteria growing better and faster and so on, we have to look at the DNA level and see what's happening to the genes and the genetic information in the DNA to know for sure. But when Lenski first started his work around the year 1990, that was pretty much impossible to do because the technology of the time couldn't fish out maybe 1 or 2 changes in DNA out of the 3 million or so nucleotides or genetic letters that comprises the E. coli genome. So all Lenski could do would be to write papers and report results that these bacteria grew faster, that sometimes their shape would change a little bit, and so on. He'd just report the results of experiments that were accessible at the time. But later on, about 10 years later, around the year 2000, the workers in his lab noticed that the strain of bacteria had lost the ability to metabolize a particular kind of sugar called ribose. And in fact, each of the 12 replicate flasks had all lost that ability. Well, it turns out that gave them a target to look at out of the vast expanse of the E. coli genome. The E. coli genome is, as I said, 3 million or so letters long, but the section that is involved in ribose metabolism is known to be at a specific place and not too big, and so they could determine the nucleotide sequence of that region, of just that region. And what they saw, what they reported it in this paper, and you can see that the title is Mechanism Causing Rapid and Parallel Losses of Ribose Catabolism in Evolving Populations of E. coli. And in another publication, they spelled it out more fully. They said that expression of the ribose and maltose regulons decreased. I'm having trouble with my pointer. Decreased after 20,000 generations. These changes are therefore beneficial because they helped the bacteria grow, but what the mutations were deletions of the genes. Turns out that another piece of DNA that has the property or ability of being able to insert itself kind of randomly at various places, inserted itself into the genes that were necessary to metabolize ribose, and in so doing, they blew it apart pretty much. They destroyed those things. And that helped. The bacteria with that mutation, with destroyed ribose genes, grew 5% faster than other bacteria. And that doesn't sound like much, but 5% in 20 generations, that has outbred the non-mutant bacteria. So the important point, the critically important point to note from this is that degradative mutations, mutations that destroy or diminish the function of a gene, can be helpful. They can be beneficial. Beneficial mutations are not the same thing as constructive mutations. After this, Lenski looked at a couple of other genes that he suspected might have had mutations in them, and the mutations were beneficial to the bacterium, to the E. coli. And here is an example of his reporting of them in the year, 2006. These 4 bars here with the letters in them, they represent 4 different genes of the E. coli bacteria, and these arrows point to positions within the genes where the sequence of DNA had changed. Usually it was one letter. Sometimes, usually one letter substituted for another. Sometimes it was missing or something else. But in the 12 replicate cultures, each one of these positions was represented at one point or another. In other words, the genes, it would be beneficial to mutate these genes but it didn't matter so much where you mutated the gene. And that is the pattern you expect to see if you are breaking or degrading the structure or the function of the gene. Because it's easy to break a gene at a number of different places because genes are complex, they code for complicated proteins, and changing something has a great chance of messing it up. Just like taking a hammer and hitting your computer at any number of places has a great chance to mess it up. So in other words, these 4 genes, and another 4 that I don't have on a slide, were helping also by losing their abilities to work, by decreasing their function. Now, in the year 2010, I wrote a, here we go. Oh, this is so cool, isn't it? Look. Notice right in the middle, whose name is that? Hey. I wrote a review paper for the journal Quarterly Review of Biology on laboratory evolution experiments, including Lenski's work up until that time. And I showed that Lenski's was not alone, that almost all other laboratory evolution experiments had shown some improvements with bacteria, with viruses, and so on. But when the identity of the mutation was able to be tracked down, it was almost always a degradative change. Sometimes it didn't affect the function, but most of the time it was a degradative change. And so I titled the paper Experimental Evolution, Loss of Function Mutations, and, quote, the first rule of adaptive evolution. And that first rule of adaptive evolution that I wrote down was this: break or blunt any functional coded element whose loss would yield a net fitness gain. Okay, that's kind of written in the jargon of a science journal, but it means simply that you should break, the first thing you should do if you want to adapt to a new situation and you're a bacterium is to break or blunt some gene, either the gene or the control region that tells the gene when to turn on or turn off. Break or blunt any gene whose loss would yield a net fitness gain. And net fitness gain just means more offspring. For some reason, evolutionary biologists call more offspring fitness. So if by breaking a gene, you can leave more cells, more baby E. coli cells, that's good. What's more, I called it I called it a rule in the sense of being a rule of thumb. That is, this is what you'd usually expect. You know, it's not a hard and fast. You don't necessarily get it all the time, but this is what you usually expect. And it's called the first rule because mutations that break a gene or blunt a gene are expected to come along first, simply because there's many more ways to break a gene than to improve one. For example, this is an illustration. It's 2 theoretical genes. If you have a gene, this bar on the bottom, and it is, if it would help an organism if that gene were broken, you could change it at any of a number of places along the way. But if you had to improve a gene by adding a new feature or changing a specific thing to improve it, there's almost always a very limited number of ways that you could do that. Just like there would be a very limited number of ways to accidentally improve your computer. So this would occur faster because mutations occur at roughly a steady rate. And if you could have a helpful effect from losing this gene, you would expect this gene to become mutated maybe 100 times faster than this one. The faster mutation comes along, and now it's available to help the organism and spread throughout the population before the other one even shows up on the scene. Okay, now many people say, how does it help an organism to break a gene? What is this? Why does it improve a bacterium's ability to grow if it throws a gene away? Well, if you think about it, sometimes in our everyday life, it helps to throw something away. For example, here's an illustration of helpful degradation. Suppose that you had a very, very pretty car, but you encountered a situation where your life depended on your car getting better gas mileage. How could you quickly change the car so it would get better gas mileage? Well, you can decrease the weight. You can throw away the doors, or you can throw out the windshield or take off the hood of the car and so on. Of course, those things are useful in many circumstances, but if your life depended on you getting better gas mileage right now, that would be the way to go. And with the bacteria in their flasks, if they don't reproduce faster than their competitors right now, then they will be swamped out. Another way is not so obvious. This is an example of a very complex chemical factory where you have a lot of things flowing into each other. And you can imagine that if one of the controls got broken, maybe this was frozen into a closed position or frozen into an open position so that it no longer worked, well, maybe the mix of chemicals would produce something new, and maybe that would help. So if you break something, maybe that will result in a helpful effect. In the year 2016, the technique of DNA sequencing had come into full flower. These days, people sequence genomes like, falling off a log. Probably many people remember, or at least people of a certain age, remember in 2000 when President Bill Clinton and British Prime Minister Tony Blair announced the complete sequencing of the human genome, which took a decade and billions of dollars. Well, these days, lots and lots of organisms have had their entire genome sequenced. Dogs, cats, mice, corn, mosquitoes, ants, you name it, including many bacteria. So in 2016, Richard Lenski's group decided to sequence the whole genome of their E. coli experiment. Not only the final result, but they had saved E. coli cells every 500 generations that they ran the experiment. So from at a point of 500 generations, they would take some of their broth with the bacteria in it, and they could freeze it away in a freezer, because bacteria don't get killed if they're frozen under the right conditions. But they do stop metabolizing, and so Lenski could go back years later, a decade later, and take some of the cells that had been frozen long ago and sequence their DNA and compare it to later generations. And so he sequenced representative DNA from each of the 12 cultures at 500 generations, 1,000 generations, 2,000, 5,000, 10,000, 20,000, up to 50,000 generations. And he published the definitive paper of his experiment in Nature. And the results of his experiment were shown in two tables. Here's table 1, here's it in Nature. Table 1 shows genes shown in the first column. Here are these abbreviations for the genes, in which only one letter has been changed. But in the third column, it shows how many different letters were changed with beneficial effect in the various strains of E. coli. And you'll notice that all of these genes had multiple places where they could undergo a mutation, and that would be beneficial for them growing in the culture. Now remember, just like the ribose gene, just like the genes we saw a couple right after the ribose gene, the four genes, this is the signature of mutations that are helping by degrading or destroying a function of the gene. So all 15 of these genes worked likely by degrading or destroying their activity. The second table was shown also in this paper, and here he showed a number of genes whose had suffered larger mutations, like insertions of a big piece of DNA, like the ribose gene did earlier, or big deletions, or other large changes in the gene, which almost always kills the gene outright. So the long and the short of it is that after 50,000 generations, which was how many at the time of the paper, after trillions and trillions of cells, the 30 most helpful mutations all degraded or destroyed the genes that they occurred in. So, what does observation show about random mutations? It's been known since the 50s that of those mutations that affect an organism, about 99% are detrimental. That is, they hurt the organism. Of the 1% that help, of even the beneficial mutations, though, it turns out now we can tell that the great majority break or degrade function. Remember, we couldn't tell that more than 10 or so years ago, simply because the technology to look at what mutations were occurring that would be helpful, that did not exist to track down mutations. The first rule of adaptive evolution says break or blunt any functional coded element whose loss would yield a net fitness gain. That rule applies always and everywhere. It is as relentless as the tide. And that's an example of this, of why I'm emphasizing this, is shown in the subsequent slides. Around the year 2010 or so, Lenski's group saw that one flask, they came in one morning and one flask was cloudier, much cloudier than the other flasks, and much cloudier than it normally was. And that's because it had a lot more E. coli cells in it than usual. And it turns out that one of the E. coli strains had developed an ability to metabolize, to eat citrate, a chemical called citrate, in the presence of oxygen. Turns out that there was a lot of citrate in those flasks for technical reasons, not as food. And ordinarily, the E. coli couldn't eat it because they couldn't bring the citrate, which is a common chemical found in citrus fruits, they couldn't bring the citrate inside the cell because the gene for the protein that acts as a pump to bring citrate inside the cell is turned off normally when there's oxygen around. And there was oxygen allowed to be in the flasks in Lenski's experiment. But one strain turned that gene on even in the presence of oxygen. And it turns out that if you see here, this region here, which is shown by the brace or so, outlined by the brace, duplicated by a genetic error, copied this twice. And now you've got two citrate genes. And it turns out this second copy here is next to a control region, this kind of brownish, yellowish region here, that is actually belongs to a nearby gene and which allows this gene to be turned on in the presence of oxygen. Since this mutant could now eat citrate, which was unavailable to non-mutant E. coli, it exploded. It gorged itself, left lots of offspring, and overtook the entire culture. Now, this result got a lot of publicity and was talked up in a lot of publications, including Nature and Science. New York Times even ran a story on it. But if you look at the mutation, I myself would think that this is just kind of it's not really a new construction. You just have a duplication of old instructions, and they get placed next to something where they weren't placed before, but nothing new was invented. So I would call this kind of a wash, an interesting result, but not really a gain of function. And many Darwinian biologists, though, thought this was just great. This was the cat's meow. And I don't want to quibble over this one mutation, but I want to focus on what happened after this mutation. It turns out that students from Lenski's lab went and graduated and became professors themselves at other schools, and they continued some of the work that they had done in his laboratory. And one group published a paper on the citrate mutants entitled Fine-Tuning Citrate Synthase Flux, potentiates and refines, in other words, helps E. coli in Lenski's experiment. And what they said was, well, metabolizing citrate in the presence of oxygen turns out to be a big change in E. coli's metabolism. It usually doesn't do that, as I mentioned earlier. They said, well, what other changes would help it in its metabolism of this citrate? And they said, well, we found one right here. They said that we found that loss of an enzyme called isocitrate lyase is predicted to be beneficial for growth on citrate. We found a nonsense mutation in that gene. In other words, they found a mutation that destroyed the activity of that gene, isocitrate lyase. Now, let me emphasize that before the citrate mutation occurred, that gene was plugging along quite happily and helping the cell metabolize in its normal fashion. When the other citrate gene was rearranged, it became beneficial for the cell to get rid of this gene, and it did so in short order. And they went on to say that, it seemed likely that these other two genes, ICLR and ARCB, are also loss of function mutations. And they had predicted by computer analysis that losing these two genes would also be helpful. And then they went back and looked at the DNA sequence of the E. coli and saw that they had suffered loss of function mutations, destructive mutations. So the point is, again, it's relentless as the tide. It's a rule of thumb, first rule of adaptive evolution. Almost always, or very frequently, loss of function mutations are beneficial. Every now and again, you have a different mutation, such as the citrate mutation. But that can't overcome the trend. The trend is always that it's faster and easier to break a gene than to construct something new. And once you get any change, such as with the citrate, the fastest way to adjust to that change is by breaking some other gene. So here's a kind of a summary lesson. Darwin's mechanism is dominated by what I call poison pill mutations. That is, they are positively selected loss of function mutations. Now, this is a whole lot worse than you might think, because since these are helpful genes, these are helpful mutations, they will spread in the population, just like the ribose mutation spread. And once it spreads through the whole population, that gene is gone forever. It's not coming back. So it's a poison pill mutation in that it's helpful, it's beneficial, but it's limiting now the species, the bacterium, in any future situation where it might have used that gene that was destroyed. And Darwin's mechanism works chiefly by squandering genetic information for short-term gain. That is, any mutation that helps an organism right now will be selected. So if it's helpful right now to throw something out, natural selection can't look ahead. Natural selection has no mind, so it'll throw things out happily. And then in the future, the organism has less and less genetic information to rely on. Well, now, I'm a biochemist. So biochemists and molecular biologists, we like E. coli. It's small, you can manipulate it, it's nice. But most people aren't as fond of bacteria as we are. So you might be wondering, well, yeah, it's interesting, but, does it work with any organism that, we're familiar with? And it turns out that, yeah, it does, including puppy dogs and all sorts of cute little animals. For example, dogs, as you know, can come in all sorts of breeds. Ones with very short legs, ones with short snouts, ones with curly hair, and ones with white patches, and so on. Turns out that the ability to sequence DNA has become so easy these days that a number of scientists have sequenced the entire genomes of all sorts of breeds of dogs and compared them one with another to ask which is, responsible for which particular trait, like short legs, like a shortened jaw or curly fur. And that's shown on this slide. And the long over here is the effect, the phenotype, the different color of the fur, whether it's curly or not, and so on. And over here is the mutation that allows that effect. And just in brief, they're also all degradatory loss of function mutations. So if you want short legs, well, let's just throw out the factor that is required for legs to grow to full length. If you want a bulldog with a shortened snout, let's throw out a factor that's necessary for facial bones to develop normally. If you want curly fur, let's mess up the gene for collagen or a gene involved in making the fur. So it turns out that breaking things can give effects that at the macroscopic level, at the normal way we see it. I was expecting to see some dogs there, but clearly I didn't. If you want even dogs that have, say, thick muscles, twice as much muscle mass as normal, that's due to destruction of a factor that stops the growth of muscles once they've achieved what is supposed to be their normal size. So you can get effects through mutations that macroscopically look pretty impressive. You see a bulldog that's got really lots and lots of muscles, but when you go and see how that was affected, it's by breaking genes. Here's another example of a creature we're more or less familiar with, the polar bear. It turns out the polar bear is thought to be descended from the grizzly bear, the brown bear, over the past 500,000 years or so. And again, sequencing DNA is so easy that the entire genomes of grizzly bears and polar bears have been determined. And they have been compared to each other because scientists are very interested in seeing what caused the polar bear to evolve. And here's the paper in which it was first reported. Population genomics reveals recent rapid evolutionary adaptation in polar bears. You go into page 5 or so, and you see that a large proportion of the mutations were predicted to be functionally damaging. Now, they can't do experiments with polar bears very easily. Wonder why. But there are computer methods where you can compare the sequence of the mutated form found in polar bear to other ones and be very confident that it's not going to work too well anymore. Oops. It turns out that this 50% figure, that 50% of the mutations is probably an underestimate because many of the genes have several mutations, and 75% of the genes that are selected contain at least one damaging mutation. So it's likely that a large fraction of these guys are degradative mutations, and they're helpful to the polar bear adapting to its environment. Now, how can that help the polar bear? Well, the most highly selected gene in the polar bear was one which was involved in the pigmentation of a grizzly bear's fur. So destroy this gene and you no longer have a pigment to make the fur brown, and it's white. Another gene is polar bears eat a lot of fat in their diet. They eat the blubber of seals. Grizzly bears eat pretty much anything, but not nearly as much fat. Turns out that breaking a gene in fat metabolism helps the polar bears digest and metabolize more fat. So throwing away genes surprisingly helps the polar bear survive. So here's a little image that I want to impress on your consciousness. This is a picture from the internet so you know it's true. And what we have here are some soldiers, and here's a convoy. It turns out that the soldiers, this is a South American country, I think, they had heard that there was a drug plant growing operation going up that road, and they had organized a group to go raid the drug growing operation. But somebody tipped off the drug growers, and they blew up the only bridge, or the bridge and the only road leading to the drug farm. Now, from the point of view of the drug manufacturers, this is a beneficial change. This is helpful to them, because they will now live another day, and maybe they can go somewhere else and set up their operation. So blowing up the bridge can be a helpful thing to do. And it turns out that that's oftentimes literally what Darwinian evolution is doing. Going back to real biology, everybody's heard of malaria. Malaria is still a killer disease, kills maybe 1 million people a year, still in 2019. And the malarial parasite is a little single-celled organism which is injected into people by infected mosquitoes. Doesn't do the mosquitoes any good either, but the malaria is using both the mosquitoes and people to reproduce. When it's injected into a person, the cell, the malarial cell, goes and enters the bloodstream, and it attaches itself to a specific place on the red blood cell membrane, the little erythrocytes, the red blood cells. And it enters into the red blood cell using that as a foothold. And once it's inside, it eats the hemoglobin in the red blood cell, and it uses that energy and material to make about 20 copies of itself. And it then bursts out of the trashed red blood cell, and each of the 20 copies attaches itself to another red blood cell. And each of them again goes inside, eats the hemoglobin, makes 20 more copies, bursts out again, and now there are 400 malarial cells. The next round there's 8,000 and 16,000, and pretty soon a good chunk of the person's blood has been consumed. Turns out there's a population of people in West Africa who are immune to malaria. And that's because, as I said, the malarial parasite has to attach to the red blood cell at a particular place. It turns out it attaches to a particular protein on the surface of the red blood cell, which is encoded by a specific gene. And this population of people has broken the gene for that protein. They have literally blown up the bridge that the malarial parasite uses to invade the red blood cell. Apparently that protein was doing something useful, but not as useful as being able to fend off the malaria. Okay, so that was the first rule of adaptive evolution. That is the quickest and the fastest way to potentially help get a change that will benefit you in some changed circumstance is to break something, just simply because there's so many ways to break things. And many of the things that impress us as really neat changes, such as polar bears or new species of finches or butterflies, can often be ascribed to broken genes. Okay, well, probably some folks here are saying to themselves, yeah, that's what you say, but what do real scientists say? And I'm glad you asked that question. Well, it turns out that my book, Darwin Devolves, in which the material is that I am discussing tonight, it was reviewed in Science magazine in February. And Science is one of the two top journals in the world, two top science journals in the world. And they reviewed it about 3 weeks before it was published. That is, they jumped the gun and they reviewed it 3 weeks ahead of time, which is often considered bad form. And they assigned 3 scientists to review the book. So they were fairly interested in it, and so you expect they'd give it a good look, and so we can learn what they had to think about the arguments. And here's the front page of the review, and so it's called The End of Evolution. And if you look here, if I can get this to stay, you look at the authors there, there's somebody and somebody else. You look at the last author, it's a guy named Richard Lenski. Richard Lenski, you ever hear of him? And when I saw this, I said, yeah, this is great. This is great. Dig into it. Tell me where I went wrong. Give it your best shot. His work was pretty much the entire chapter 7 of my book. And he is a molecular biologist and an evolutionary biologist. And he's pretty much the best scientist in the world to be able to evaluate these arguments. And in the very first paragraph, they didn't really say what the book is about. He writes, in 1996, biochemist Michael Behe introduced the notion of irreducible complexity, arguing that some molecular structures could not have evolved because their functionality requires interacting parts. This claim excited creationists and remains a central plank of the intelligent design movement, despite being rightly rejected by a U.S. federal judge in 2000. In Darwin's Evolve, he continues his chaotic efforts to overturn modern evolutionary theory. So he wants to be fair. He doesn't want to influence his readers overly much at the beginning. He just wants to tell them what the argument is. Now, that is, this is terrible. I mean, I've never seen a review quite as uninhibited in its disdain and attacking of a book that is going to be reviewed after the first paragraph. So he then goes on to talk about the actual arguments, and that many of the, as I wrote, the most mutations that help are loss of function. And he writes, yeah, there are indeed many examples of loss of function mutations that are advantageous, but Behe is selective in his examples. He dedicates the better part of chapter 7 to discussing a 65,000 generation E. coli experiment, emphasizing the many mutations that arose that degraded function. An expected mode of adaptation to a simple laboratory environment, by the way. It wasn't expected before the book came out, let me say. While dismissing improved functions and deriding one new one as a sideshow. Full disclosure, the findings in question were published by co-author Richard Lenski. Okay, so he says, yeah. He says, sure, there are these loss of function ones, and they help, but I don't emphasize enough the ones that improved functions. And the one that improved functions, the one he meant, is the citrate mutation. And actually, if you read the book, I say, this was a very interesting result, very interesting. I said, but if you look at the trend, his students went on to show that, in fact, it was followed up by more and more loss of function mutations. And I did say that mutation, the citrate mutation, was a sideshow, and I didn't mean to hurt his feelings by calling it that. It seems that he took it that way. I just meant to say that it's a sideshow in the sense that the great trend is to degrade genes and loss of function. And this was I think this was the third paragraph or so of the review. And the rest of the review, he doesn't say a word about his own experiment, his own life's work, even though that's exactly the level of biology that I'm concentrating on, the molecular changes in DNA. His is the most prominent, the most celebrated investigation of exactly that, and he does not deal with it. They also go on to say, let me skip. Okay, Behe also ignores the fact that some of his prior arguments have been dismantled. He includes a lengthy appendix that argues that the blood clotting cascade is irreducibly complex, for example, but fails to mention Kenneth Miller's simple, elegant scheme for its stepwise evolution. So some of you may know that in my first book, Darwin's Black Box, I argue that a number of systems in the cell are irreducibly complex and therefore big problems for Darwinian evolution, and one of them was the blood clotting cascade. And later on, a professor of biology named Kenneth Miller from Brown University wrote some counter-arguments to it. However, if you look at the citation here that Lenski has, 3, and if you look it up at the end of the article, the citation is to a chapter that Ken Miller wrote in a philosophy of biology textbook. But it is a duplicate of a chapter that was published 5 years earlier in another philosophy of biology book, which was called Darwin versus Design, and was published by Oxford University Press, I believe, a very prestigious academic publisher. And in that, Ken Miller did say something about the blood clotting cascade, but he was mostly concerned with the flagellum. He only wrote something like a few sentences about the blood clotting cascade. But if you look at the table of contents, you'd see that there were other contributors to this volume, and one of them was myself, in which I addressed and answered all the criticisms that Kenneth Miller had put in his chapter. And yet Lenski doesn't even mention that, and he leads the readers to think that I just ignored the criticism and did not engage any of these things. In fact, I've written tons of stuff over the past 25 years. I've engaged critics left and right, and yet a problem is that whenever a critic starts to argue, they don't refer to the conversation, but like Lenski, they think that the world is still new and that the arguments that they have read have not been addressed. What's more, if you look at Kenneth Miller's book and even his web page, you'll find that the extent of his argument on the blood clotting system is about a paragraph in length. It is terminally vague and hand-waving, and he has never published anything in the scientific literature about that. What's more, Kenneth Miller does not work on the blood clotting system himself. He just reads papers on it. People who do work on it, like a man named Russell Doolittle, who was a professor of biochemistry at the University of California, San Diego, I have shown numerous times that they have never published, and they've published mistaken arguments that clearly show they don't know how random mutation and natural selection could build a blood clotting cascade. Okay. Okay. That's just what I said, but I just told you what I said. And here's the final argument. No, it's kind of the penultimate paragraph or so of the review. He says, well, yeah, I talk about devolution, but acceptation also challenges Behe's notion of devolution by showing that loss of one function can lead to the gain of another. The evolutionary ancestors of whales lost their ability to walk on land as their front limbs evolved into flippers, for example, but flippers proved advantageous in the long run. And that is the extent of his argument. He's saying, yeah, sure, the limbs of whale ancestors had legs, but look, whales have flippers. So look at what Darwinian evolution can do. It can substitute one for the other. And you say, wait a second, where is the evidence that Darwinian processes did this? And there's nothing. There's no citation. There's nothing. The whole point of the book was to argue that Darwin's mechanism, I don't argue the point of evolution in the understood as common descent. I say, even if that's the case, Darwin's mechanism of random mutation and natural selection cannot account for these changes. And yet Lenski doesn't even grasp that argument is being made. He just says, look, here's this, look, here's that, what's the problem? And I don't mean to be mean to Richard Lenski, who's done wonderful work. His E. coli experiments are just great, and they show us things about evolution that we would not have known otherwise. But like many scientists, they take something for granted and don't know that they're taking it for granted, and when challenged on it, can't give much of an answer. Okay, I'm just seeing how much time is going by here. Okay, so yeah, the last paragraph of the review says, ultimately Darwin devolves fails to challenge modern evolutionary science because, once again, Behe does not fully engage with it. He misrepresents theory and avoids evidence that challenges him. Now, if any of you have taken a psychology course, you might have come across the term projection. And that is where, of course, somebody ascribes faults to another person that is actually the person's faults themselves. And it's my opinion that, in fact, it's Lenski and his co-authors and the entire evolutionary biology field that misrepresent theory and avoid evidence that challenges them. Okay, we have some more like that, but time is marching and I want to leave a lot of room for questions, so we'll just skip over that. Oh, that's really good. Oh, yeah. Oh, too bad. Okay, and I should let you know, of course, you can tell that I've got some critics of my position, and all of these things can be found by my responses to the criticisms can be found at various places. One, my faculty website at Lehigh University, and the website of the Discovery Institute, and also the website of the new book, darwindevolves.com. They have all of my replies. If you want to read the original criticisms themselves by the other authors, they're easy to find. Just type my name in some web browser, plus any common swear word, and and you'll get lots of criticism. So thanks very much for your attention.
We'd like to invite, if anyone has questions for Dr. Behe, we have microphones here in the front. Come on up and ask a question. And I'll just start things off by saying, Dr. Behe, why should we care? So what is this? So most of us here aren't molecular biologists or biomechanical, biochemical, whatever you are. And so why is this topic important?
Well, there's a number of reasons that people outside of just evolutionary biologists and biochemists should care. One is practical reasons are, if you're involved in wildlife preservation or species preservation, and you want to understand what is affecting the ability of species to adapt to changes and so on, it's very important for you to know that, in fact, the best way, the fastest way they adapt is to lose genes, to lose genetic flexibility. It has medical ramifications too, because many diseases, many mutations in humans have been selected as a response to genetic diseases, and overwhelmingly they are broken genes. And so that has implications for medicine, maybe what to do about it is a different question. But most importantly, I think, is that we all want to understand how our world works. What is going on? Everybody at some point when they're 5 or 6 years old says, wait a second, how'd this all get here? Where'd this come from? And for the longest time, we've been assured by the respected authorities in our culture that it's pretty much just been chance. That, the same forces that, cause the wind to blow or cause the sun to shine, things like that are the reason you're here, and it's no big deal. But through the very progress of science, you should remember that from antiquity up until the year 1800 or so, everybody in the world, smart folks, everybody thought that life was designed, because we see how it's put together, and that's how you recognize design, when you see a purposeful arrangement of parts. It's only been in the past 150 years or so, an aberrant period of history, that people have been assured or taught or thought that, in fact, life is not designed. And that, now, though science, through the progress of science, as we've reached deeper and deeper and deeper, we see that, in fact, we are, I think, what's the phrase? Fearfully and wonderfully made. Even the foundation, the molecular and cellular foundation of life is astoundingly complex, extremely elegant, and it's getting more and more so the more and more we learn. And we were told that, this could be imitated by randomness, random changes plus this selection, kind of a sieve. And now we see strong evidence that, like most of Darwin's skeptics have thought since 1859, Darwin's mechanism might work around the margins of life, but it most often does so by degrading creatures. And people in the 19th century thought in terms of degrading creatures. Now, since we've discovered the molecular basis of life, we can see that it helps by degrading the very genetic patrimony that a species has inherited. And so that we and every other species are spending our genetic endowment whenever we have to adapt to a new set of circumstances. So it's got practical, it's got scientific, it's got philosophical, maybe even theological implications, and I think there's probably something in there for everybody to be interested in. Anybody else interested?
Dr. Behe?
Yes.
I guess I felt like there was times when you were saying most of the time this was happening, that most of the time it was degradation causing these mechanisms. Does that leave any room for any times where something was actually creating function, creating some new feature?
And so,
I guess why was that language used is kind of the question I was asking myself.
okay. So the question is, can it make a new function? Can random mutation and...
I guess I feel like it was like everything but 100%. It was like 99% of the time.
No, I would say, oh, 3/4 or so, something like that. But, it's you've got to make some fine distinctions. And I tried to do that in that paper I wrote on the quarterly review of biology that I had up on the screen. If you, at the molecular level, DNA contains coded information. You got genes that code for proteins. And you've got control elements whose very sequence codes for it to be turned on or turned off or something like that. You've got proteins that have sites on them that can be modified. And in my paper, I call all of these things coded elements. So the question then arises, does an organism adapt by gaining coded elements, which I called gain of coded elements, or loss of coded elements, which I called loss of functional coded elements, or something else, something in between? And I would estimate that 3/4-ish are loss of function, and the very great remainder of them are neither gain nor loss, but either rearrangements of particular things or duplication of regions that I wouldn't count as new functional coded information.
And then, sorry, there's a line behind me, but the other question I had is CRISPR is kind of an interesting thing that I've heard about where you're inserting instructions like you can use bacteria, functions from bacteria to do that. Is that a mechanism for inserting functionality or other structures into things? Is that a mechanism
Sure,
that
okay.
is being considered?
Okay, yeah, good. There is this technique, probably most people have heard about it, called CRISPR, where folks who know how can use it to place pieces of DNA in a particular organism, or they can use it to destroy particular regions of a gene. Sure, using CRISPR, a scientist could insert new genetic information into an organism, but of course that would be intelligent design. If I took a gene, there's a gene for something called green fluorescent protein, which literally fluoresces green when light is shown upon it. If we put that in, say, actually some people did put it in the genome for a zebrafish, or I think, and shone light on it, and you had a green fish. And some people wanted to sell this in pet stores and so on. Well, yeah, I guess that's certainly gain of information in that lineage of that fish, but it's intelligent design once more. So it doesn't affect the question of where did the machinery that constitutes CRISPR come from, and how is it regulated in life, and when you develop something like that in the complete absence of a guiding intelligence. And I would think the answer is pretty much no for that. Yes, sir.
So my question is, oh man, I just lost it. Give me 1 second. I'm sorry. Basically, it's very difficult for a mutation to be beneficial to a gene, and most of the times it's breaking a gene. Is there an experiment that could be run to determine just how fast that's swamped out? Just how fast the destruction of useful genes compares to the once in a while beneficial change to a gene, and just see how fast it deteriorated? How would you go about running an experiment for that?
Well, that's a good question. What you have to do is pretty much let things grow and see what happens, which is what Richard Lenski did.
If I may throw one caveat in there, other than Dr. Lenski's work, because I think that's a very good experiment to cite, but beyond that, in different cases, what other methods, what other experiments?
Well, a colleague in my department at Lehigh does work similar to Lenski, but using yeast, which is a eukaryote and has a somewhat bigger genome and so on. And they get pretty much the same thing. They get beneficial mutations, and they're all loss of function mutations. So I don't think in either case, my colleagues or Lenski's or anybody that I'm aware of has a new constructive coded element been made. I think once for a virus, in a virus that replicates, very, very fast, and it's a special case that I talk about in that review paper. But overwhelmingly, it's either loss of function or just kind of rearrangement of stuff that was already there.
So it's not comparing like an 8-to-1 ratio, it's comparing an 8 to basically 0
I'm
ratio.
sorry?
Basically saying that the amount of degradation that takes place is so fast that comparing it to useful things is utterly negligible. You can't even say it grows 100 times faster, 1,000 times faster. It's just utterly negligible.
Well, yeah, but of course you can't say, with Richard Lenski's results, for example, in his landmark paper, he talked about 30 different genes. Okay, so 30 is a relatively small number. You can't say that it's 1,000 times, 1,000 times the number of constructive mutations if you only have 30 examples to start with. But in every case we've seen so far, we haven't seen anything. So I think even 10 to 1 would be enough to conclude that things are inevitably headed downhill. You don't need it to be, 1,000 or 1,000,000 to 1.
Thank you.
Yes, sir.
Hello. I was reading this book written by Michael Shermer. He is not on your level. He doesn't deal with the kind of things that you deal with. Michael Shermer.
Oh, Michael Shermer,
He
yeah.
wrote a book. He's an atheist.
Yes, yeah, I know him, yes.
This book is riddled with fallacies. It's actually a great exercise in being able to pick them out. However, he did attack a couple of your arguments. You made two arguments, one for the flagellum and the other one for the human eye. I felt like he actually dealt with the eye, the human eye, being it not being inconsistent with evolution. Do you still affirm that the human eye is a proof for proof against evolution or for intelligent design? And can you explain that by the molecular side of things?
Well, there are two things. What I think and what has been shown so far, what can be demonstrated. I don't think Darwinian mechanism could do anything, remotely as complex as a vertebrate eye. In Darwin's Black Box, I wrote about the eye relatively briefly, and I said that it had long been argued over, and talked about William Paley, and then Charles Darwin himself wrote about the eye, and Richard Dawkins later. used the same argument as Charles Darwin. And I said, all of these arguments, are beside the point because they're at the level of organs and physiology. But we now know that the foundation of life is at the molecular level, which Darwin knew nothing about. Neither did Paley, but Dawkins does, and yet he does not address the problem at the molecular level. And I said, what happens in vision? In vision, a photon of light first hits a small molecule called retinal. The retinal has a shape like this, but when the photon hits it, it isomerizes like this. And it's a switch that, when it changes shape, changes the shape of the protein to which it's bound. That change in shape allows another protein to stick to an exposed site that hadn't been exposed before, turning that protein on. It's called transducin. That, in turn, affects another protein, which affects another, and so on. And I said, nobody's ever tried to explain this. And that was in 1996. To this day, nobody has still ever tried to explain how these molecular systems could have been built. The problem is that when you talk about eyeballs, Darwin's classic argument was, well, okay, we know the vertebrate eye is really complicated, but suppose we start with a light-sensitive spot, and then it kind of curls up, and then it gets filled with a jelly like a lens. That's not so hard. Well, it's not so hard because it's a cartoon. A little light-sensitive spot contains, dozens and hundreds of complex proteins, which are themselves machines. And what would cause a flat cell to curl up? Why should it do that? There are dozens of complex proteins involved in shaping cells, and they interact with each other in very specific ways. And none of that has, people just wave hands. I heard somebody mention an analogy the other day, which I thought was clever. And they says, if you think of Harry Potter's wand, what can Harry Potter's wand do? You say, well, in the real world, you can use it to stir your coffee, or you can use it to, smack the dog or something like that. But in the movies, you can, do anything. Darwin's theory is like that. In your imagination, you can do anything with it, but you can't even stir coffee with Darwin's theory in everyday life.
So to prove evolution true, one needs to prove it on the molecular level.
Yes.
Correct? Okay.
Exactly. That's the critical lesson that I hope everybody takes home, is that in order to evaluate any claims about Darwin's theory, you have to look at the molecular level of life, just because that's where the action occurs in biology. Yes, ma'am.
Hi, so I am actually a major in biochemistry right now. I'm an undergrad. And I guess my question, all of my teachers in every class I've ever had with biology, they all talk about evolution like it's irrefutable fact. So some of the evolutionary theories are easy to pick out, like when they say that a cell engulfs mitochondria and just decided not to eat it, so therefore eukaryotic cells have mitochondria. That's easy to pick out, but sometimes they're very tricky about it, like these two bacteria can share genetic information, therefore our genome is made up of mostly viral information, therefore evolution exists.
Yeah.
So I guess my question to you is, how would you go about sifting through what is actually fact and what we actually should learn for biochemistry versus what is just theory that they're trying to teach as fact?
Okay. Okay, well, you should assume that whenever anybody says, this evolved from this because this happened, it has absolutely no evidence backing it up, other than the fact that this might look like that somehow, or the sequence of this gene might look like the sequence of that gene. But Darwin's claim to fame is not that he saw relatedness among organisms, but he thought that there was that was an unintelligent process existed that could explain even the most complicated biological system. I'm afraid it's difficult. I was taught the same thing you are being taught now, and it was only reading outside of the general literature that I realized that there were questions to which I did not have answers. And when I tried to track them down, I saw that it was kind of like a Potemkin village, just a facade of knowledge. Unfortunately, yeah, pretty much you can, in the biochemistry is best at telling you what's happening now. How do cells work right now in extant organisms? The speculations about how they came about, they're pretty much worthless. They're there, if you want to be charitable, you could say they're there in textbooks to kind of train minds in how to think about a subject. But they're training them in the wrong way. So I don't know if that's a good reason or not. In most biochemistry textbooks, at least up until 1996 or so, didn't say anything about evolution. I had a chapter in my first book, Darwin's Black Box, surveying the literature, and I surveyed about 20 different biochemistry textbooks and showed that none of them had anything substantive on evolution. Every now and again they would say something like, isn't it wonderful how evolution has provided this system for us? But they would never treat it seriously. And lately, since the publication of the book and since people have called them on it, they've tried to be more substantial, but they still don't get it that, you got to show that random mutation and natural selection could explain this stuff. You can't just show that, say, the gene for myoglobin is similar to the gene for the alpha or beta portions of hemoglobin, and it's a really, I wish I could be more positive, but you as an individual, you just have to read more widely. There's lots of skeptical stuff. Even, outside of intelligent design, many biologists, when they're not being interviewed when they're not speaking in public, I would guess at least a third, maybe a half of biologists don't think very much of Darwin's theory at all. They think that there must be something else going on to explain life. But they, can't say that out loud. But there are websites and books by run-of-the-mill or real respected biologists, not renegades like us, that say Darwin didn't, he missed the boat, he didn't get anything right, he's utterly wrong. Look at my new theory. My new theory gets it all right. And you look at the theory, it's not so good, but you're left with agreeing with him that Darwin didn't get anything right. I guess the short of it is you just have to read more widely. You can't depend on textbooks or explanations from your instructors, especially if you sense that they are committed to this point of view.
Thank you.
Yes, sir.
I just wanted to jump back to, you mentioned the dog genetics, and you talked about short legs versus long legs, curly hair versus...
Could you speak up?
Yeah, you gonna hear me now?
Yeah.
I was wanting to refer back to the slide you showed us on the genes in the dogs, the long legs versus, you don't need to bring it up, the long legs versus short legs, curly hair versus long hair. Do you know if anyone has looked at, in that paper you said that they found that there was like a mutation in, some part of the protein that makes the hair curl. I'm assuming then that you could look at if the animals are heterozygous for the trait, you could look at the dominant, you could look at the recessive, and you could compare the sequence between the two. And if the sequence was almost the same, you would assume, okay, well, this is probably a loss of, function in one of these genes. Do you know what the ratio, if you look at the total amount of genetic variability that there is within, an individual, how many of those look to be a loss of function in what otherwise would have been all straight hair, for instance, in the dog, versus a gene where you've got genuinely new information. So I'm not familiar enough to...
Okay, so... there we go. Okay. Okay, and natural variation, say in human populations or other species populations, what's the proportion between loss of function and new genetic information? It's hard to tell. Probably a real good chunk is loss of information or degradation. And that's one reason why you don't want to marry your cousin, because if you have the same genes, and oftentimes people carry damaged genes, and if you marry a close relative and the offspring has 2 damaged genes, that's terrible. And I can't think of any natural variant where some new regulatory element or some new gene or part of a gene, or I can't think of any in the human population that is active, that people have tracked down. You might say, well, there might be one over here, or there might be one over there that we've missed. Maybe so. But typically you get things like, the mutations that have grown in populations in response to malaria. There's not only the sickle cell mutation and that other one I talked about where that gene for the protein on the surface of the red blood cell is destroyed. There's also things that cause a disease called thalassemia, in which genes for portions of hemoglobin are destroyed. Duffy antigen, glucose 6-phosphate dehydrogenase is destroyed. They're all degradatory ones. You might have heard, some folks might have heard that a while back, that there's a population in Europe that has resistance to the HIV. They don't get HIV. And it turns out that, this was portrayed as a real, we're mutating into Superman or so. But it turns out that's the loss of a similar, of a protein on the surface of a cell to which the virus attaches. There were stories, I have all of these things in my book. There were stories in the New York Times a few years ago, one in which it said, these population of Amish in Pennsylvania, they have low cholesterol levels, and what's more, they can drink milkshakes and their cholesterol doesn't go up at all. There's no history of heart disease in their lineage and so on. They tracked it down, and it was a protein involved in lipid metabolism, loss of function. People in Iceland, they tracked down a resistance to type 2 diabetes, and so these folks don't get type 2 diabetes. It was a loss of function mutation. There's a horse, has a special gait that makes it smoother to ride, and it is bred by horse breeders because riders like to ride it, and it's the destruction of a gene involved in nerve tissue. So those are all over the place, and I'm afraid I can't think of one that is a new gain of function.
So I'm not thinking gain of function. I guess I'm not sure what your beliefs are on origin of how we started out. I guess what we look like when we started out. But I guess I posited kind of the ability to have rapid speciation if you had a lot of genes to choose from to start with. But if we find that most of the genes, and when I say genes, I'm talking like the dog example, curly hair versus straight hair. If most of those are more similar to just single nucleotide polymorphism sorts of mutations where you 99% of it's the same, you have one little break in it, that would seem to suggest that most of the variability that you see is actually a result of degradative sorts of processes, versus if you were to say, well, I think there was rapid speciation X number of years ago, you would still expect to see genes in the population where the dominant versus the recessive, for instance, would both be very dissimilar and have a lot of complexity.
Okay. I'm not quite following it. I'm sorry. But yeah, I'll talk to you afterwards, okay? Yeah, yes, sir.
Hi, Dr. Behe. I was listening to a recent podcast on intelligent design the future.
Uh-huh.
And the subject was on orphan genes. Orphan genes.
Orphan genes, okay.
And they mentioned a couple organisms that had a particularly high amount of orphan genes. And one of them happened to be E. coli. And then another happened to be bees. And it was specifically related to how they communicate. Apparently they tied their genes to the ability to communicate, which were orphan genes. And I thought that was super interesting, but I guess my question is there a link between these organisms are both very, very important to human thriving, just being here, bees and E. coli. And I was wondering if there was maybe an anthropic type principle that indicates that, or that shows that the more important a species or organism is to humans, the more orphan genes we would find in that organism. Is that something that's been investigated or looked into?
If it has, I don't know. I mean, or for the audience, an orphan gene, it's kind of a play on words. ORF in biology lingo stands for open reading frame, which means essentially a stretch of DNA that looks like a gene, but people don't know whether it's used by the organism. They don't know what it does. They don't even know if it makes RNA, if it's expressed or not. And so they call them orphan genes, and they are not found, they're also not found in other species. So that one species or one branch of biology will have genes that are essentially unique to them. Another branch will have other genes that are unique to them, but both will have also genes that are common to pretty much all species. And people have been scratching their heads about these for a while, and they figured that they would decrease the more we knew about DNA and the more we knew about genes from different organisms, but they haven't. Even though the number of species whose DNA has been sequenced has gone up exponentially, they still have, up, say, 20% of their genes seem to be restricted to certain groups or have unknown functions. And I think the question was, is this more prominent in species that are of use to people? And I haven't heard anybody make that correlation before. It may be the case, but I don't know. There are folks at the Discovery Institute, a man named Paul Nelson in particular, who are very interested in the topic of orphan genes and have some research projects going on in that area. So people are working on that questions like that, but it'll be a while before they reach any definitive conclusions, I'm sure. Yes, sir.
Oh, yes, Dr. Behe, thank you for speaking tonight, first of all.
Okay, let me just say that, just so I don't, just so people feel free to leave if you'd like, or after these two gentlemen, then we'll call it a night.
Okay, just real quick. Actually, I just wanted to I don't know. Regarding the question of the gentleman that was standing in front of me, actually, I'm just trying to understand your answer, or maybe clarify, or just get some understanding on it. I guess I'm wondering if, from your answer, if you're saying then that, for example, that if there had not been genetic degradation in the past, are you saying that if, like, there had never been any genetic degradation in the past, are you saying that all humans would have basically the same genome in that case?
I'm not sure that I said that. I didn't say that.
Okay, so you don't say that, because I
no.
felt like that was what his question was getting at, because he was saying is all the variation, like why one human's genome is different from another human's genome, is that all because of genetic degradation? And then he was asking what percentage of the variation would be because of degradation, and what percentage of the variation would be due to some difference from some other cause.
Okay, well, mutations happen, okay? It turns out that people, a new baby is born with about 50 mutations that his parents did not have. 50 new mutations per generation, per person. That's just because, although the machinery of the cell is really cool, it's not perfect. Nothing in this world is perfect, so mistakes are made. In order for an improvement to occur, you would have to select for that in a Darwinian mechanism over generations. So in order for one of those mutations or some of those mutations to contribute to improving the genome, it would have to be selected over time. I do not know, as I said before, I don't know of any examples of new functional coded elements in the human genome. There are ones which gunk things up, which break things, and then there are ones that look like maybe they don't do anything, they're neutral.
Okay.
But I don't know of any that make new proteins or make new control elements.
Yeah, I guess maybe the answer is in there from logical inference. Thank you for,
Okay, you bet.
Yeah, I'm just trying to summarize this for myself, for my own teaching and things like that. So Darwinian evolution would tend to suggest that if you look at a group of mechanisms over a very long time or a very high number of generations, then they're subject to normal random mutation, mistakes in replication, stray radiation, whatever, and environmental stresses that you would expect the simple to become more complex because information is gained. But what you found is essentially just the opposite. You have a very, very complicated mechanism that sheds complexity or certain aspects. You have a complicated mechanism that has a complicated mechanism that decides what complicated mechanisms to destroy or to delay. That's what your findings show. So complicated mechanism that's executing a complicated plan to decomplexify certain portions of itself in order to benefit the mechanism, the organism, which is kind of exactly the opposite of what Darwinian theory suggests that we should find in something like Lindsay's experiments. Am I summarizing that correctly?
Yes, that's correct. That, in fact, a lineage starts out and it's doing fine in its environment. Maybe some species go, or some members of the species go to the right or to the left, and they encounter a different environment, and some random mutation comes along which messes something up, and somehow that helps them survive in their new environment. And then that happens over and over again. And with that, you can have, it's really actually nice because you can have species branching out and filling all sorts of niches, all sorts of different specialized environments. And so you fill the earth, you fill the environment, but you're doing it by losing information, not by gaining information. If you think about the polar bear, that's cool. You know, polar bears are really cool, and they're adapted to this really tough environment. But if global warming warms up the Arctic and you start to get forests there, it's not going to go back and get brown fur, and it's not going to be eating berries and stuff. It's coming to a dead end. It's kind of like, you know, some rich kid who has inherited a bunch of money, and, you know, he goes and spends it here, and, you know, and then he goes and spends it there, and eventually he runs out of money, and he's stuck where he spent his last dime. He's not going anywhere. And I remind people that it's known that, you know, 99% of species that have lived have gone extinct. And if you think about it in these terms, that is that species lose stuff in order to adapt. Then if they've encountered enough changes where they have to lose this and then lose that, eventually they are running out of options, and they might just, the next change that comes along, they might go extinct instead of adapting. Okay. Thank you.
We would like to thank you, Dr. Behe, for being here tonight. Yes. I don't know about you all, but I'm going to attack him and ask him more questions. So cue up. Thanks for being here. It's so good to have you. It took a lot of work to get you to Colorado. And it didn't snow. So we're very excited. So if you brought your Behe book with you, I'll bet you can convince Mike to sign it for you. And yeah, I think we'll head out to the lobby so the staff can clean up the room and get it prepared. So thank you all for coming. Don't forget to check the book table in the back and pick up a brochure and sign up for our email list. Thank you all.